Hello and welcome to this Bioprocess Insights webinar. I'm your host, Leah Rosin, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Mats Lundgren from GE.
Thank you for the introduction and a warm welcome also from me to this webinar about vaccine process economy. But first I will give a few words about myself. I work as a Customer Applications Director at GE Healthcare Life Sciences based in Sweden. I have worked for more than 25 years in the field of vaccinology. I have a PhD from the Karolinska Institute in Stockholm and a postdoc training at the Imperial College School of Medicine in London. In my industrial career, I've had positions as a scientist, team manager, and vice president at Pharmacia, AstraZeneca, and some smaller vaccine companies. In my current role at GE, I work across different vaccine application projects focusing on new technologies and manufacturing solutions. So this is the outline of the presentation. I will first discuss the need for updating vaccine processes. After that, I will continue with some reports and presentations from users of single-use process technologies. We will move on to 2 case studies describing the economical implications of introduction of single-use technology upstream and using a more modern technology downstream. And finally, I will conclude the presentation. So, why do we want to discuss updates of vaccine process technologies? Well, manufacturing costs are actually very substantial for vaccines. In this example, you can see that manufacturing cost actually represents more than 50% of the overall cost of a vaccine. So thus, the improvements in this area are really important for profitability in vaccine companies. And as we all know, vaccine companies are often under quite severe cost pressure. So there is a need for updating vaccine processing, and here you can see some of the reasons for this. First of all, you have often unfavorable process economy, and this can be due to low yields in your process. Also, in some cases, very long process times and very labor-intensive processes. Also, in many cases, dedicated facilities were built for these processes in the era of the big stainless steel facilities. Also, processes were developed decades ago. This means that a lot of old expression and purification technologies are used, such as old and very difficult to grow cell substrates or even eggs. Sometimes also limited purification in the process. And for this reason, you need a significant expertise, which can make it difficult to do, for example, a tech transfer. Also, we have processes that are difficult to scale up due to some inherent problems with the process technologies that are being used, such as roller bottles, centrifugation, and also big fixed stainless steel installations. And finally, we have also an increased regulatory quality requirements in the industry. One of the major regulatory concerns in vaccine manufacturing is really the potential risk of cross-contamination that comes with, for example, inefficient equipment cleaning procedures, also open handling of material. And also you can have batch variability that can be of a concern. Also the use of animal-derived components such as serum or animal-derived trypsin also means potential introduction of adventitious agents into the product. So why do we think that disposables are suitable for vaccine production? Well, vaccines in comparison to other biopharmaceuticals are often produced in relatively small batches. And the smaller scales are often in the range suitable for disposable equipment. Many manufacturers also produce various vaccines in campaigns. And by using single-use, you can actually facilitate multi-product manufacturing more easily than with traditional stainless steel formats. Also for pandemic preparedness, single-use technologies can really speed up development and scale up of your product. And as the theme of this presentation, cost pressure on vaccine manufacturers call really for new approaches in production and the technologies that are used in manufacturing. And finally, as we have said before, one of the major regulatory concerns are potential cross-contamination, and single-use technologies and closed systems can reduce this risk quite significantly. However, there are many reasons to use single-use technologies, but the effect on process economy must be studied quite carefully before you can implement it. So we will now look at what users of single-use technologies report in terms of process economy implications when they have introduced single-use into their manufacturing facilities. So we have compiled a lot of data actually over the last 5 years from various publications and conference presentations that are covering process economy for single-use technologies. And in this table you can actually see 8 different companies ranging from large multinational vaccine companies, smaller vaccine companies, large multinational pharma companies, large biotechs, smaller biotechs and CMOs, and what they report in terms of their economical implications of introducing single-use technologies. First of all, you can see that as expected, you have an increase in the amount of consumables that are being used, and it ranges between 25% up to even, from 50% up to 250%. So that's of course quite a lot. However, you see that facility costs are actually much lower, ranging from 25 up to 75%. Also, the facility footprint is reduced quite significantly, as well as the labor required in the facility. What is quite interesting also is that time to build the facility is reduced quite a lot. You can see reports from 25% up to 50%. Also, the turnover time between batches is reduced, as well as water and energy consumption. And we also see one company here reporting quite a large capacity increase of 30%. So all in all, cost of goods can actually be reduced quite significantly. And you can see that it ranges between 25 and up to 67%. So it's really significant savings you can have when going into single-use manufacturing. So if you want to summarize this, we have seen that users of single-use technologies report facility cost savings and footprint reduction, also savings in terms of facility build-out, equipment cost savings, labor cost savings, cycle turnover time savings, and also savings in terms of water, chemicals, and energy. We will now turn over to some studies that we have done internally. But also these studies are based actually on data coming from discussions with our customers. These calculations were made using Excel mainly, but we have also used other software tools. We have done a very detailed scheduling of the investigated unit operations. And the calculations include capital investments in terms of hardware and facility, also cost of labor, cost of disposables, raw materials, water for injection, steam, etc., as well as cost of waste. In the first study, we will compare the use of microcarriers in stainless steel and single-use bioreactors for expansion of adherent cells that will be used for virus production. And in a conventional setting using our standard microcarriers, there are a number of preparation steps that need to take place before the microcarriers can be inoculated with cells, as you can see to the left in this slide. For example, you need to sterilize the carriers before use. If you use a stainless steel bioreactor, this can be performed inside the bioreactor. However, if you use single-use bioreactors, this has to be done in a separate vessel, as the plastic bag cannot withstand the heat during sterilization. So in response to this, we have developed pre-sterilized microcarriers that can be added directly to the bioreactor, and thus you save a lot of preparation time. So the pre-sterilized microcarriers really enable the use of disposable bioreactors in an efficient way. So in this case study, we have looked at cell expansion from cell bank vials via T-flasks and cell factories, up to 50-liter bioreactors, 200-liter bioreactors, and finally 2 times 500-liter bioreactor volumes. As I told you previously, the use of pre-sterilized microcarriers will save a lot of time. Another benefit if you are using single-use bioreactors in combination with pre-sterilized microcarriers is that you don't need a steam-in-place installation in your facility. And this is, of course, quite a big saving, both in terms of money and footprint. So in this slide, we have looked at a number of batches that can be produced at maximum capacity using pre-sterilized microcarriers. And the manufacturing capacity increased by 64% by using single-use bioreactors. And this is, of course, a very significant increase if you need maximum output from your plant. And in this slide, we also looked at the relative cost per dose for the upstream part of the process. And this is also reduced when using single-use bioreactors, as you can see to the right. We have actually also looked at the relative costs for generation of a certain amount of cell substrate. So if we assume that the virus productivity per cell is similar, which is reasonable, this will give us an indication of the upstream cost using different cell expansion technologies. In this diagram, we have compared first stainless steel bioreactors using standard Cytodex with single-use bioreactors using pre-sterilized microcarriers. This is also compared to with roller bottles or cell factories. As can be seen here, single-use bioreactors with pre-sterilized microcarriers is the most cost-efficient alternative, as you can see here. In this example, the roller bottles and the cell factories are based on manual handling. And of course, you can use robotics for handling of rollers and cell factories, but this also means quite a substantial investment. So now we will turn over our interest into the downstream side of the process, and we will look at the process economy evaluation using core bead chromatography technology. One of the most common techniques used in vaccine purification is size exclusion, or also called gel filtration chromatography. This has been used for very many years and very successfully. And with this technology, you separate components based on size. Size exclusion is a highly efficient separation technique. And the only sort of challenge with this is that it requires quite large chromatography columns when it's used in industrial scale. And due to the column size, this means that it is somehow difficult to use single-use chromatography columns, as you are restricted to the size of the column. So another alternative for some of these processes is actually to use core bead chromatography. And core beads are very sort of interesting technology, I think. They have an inert surface with pores, and inside the bead we have a very strong ligand that will bind almost anything that enters the beads, such as impurities. And depending on the size of the pores, you can bind smaller impurities such as host cell proteins or fragmented DNA, while leaving, for example, your virus target on the outside of the bead. And actually what happens is that the virus will go in the void of the column, and it will actually pass through the column, whereas the impurities are captured by the bead. And one advantage with this technology is that it's much easier to scale because the columns can be quite small. And you can actually use single-use chromatography columns. In many cases, the purification performance of core beads is the same as size exclusion chromatography, as you can see here. For example, the antigen yield is very comparable between the two technologies. Also, host cell protein reduction is very comparable also. A very interesting property of the core beads is that you can reduce ovalbumin very efficiently, even more efficiently than compared to size exclusion, which makes it a really attractive solution for purification of egg-based vaccines. But needless to say, the suitability of the core beads compared to size exclusion has to be tested in each case. However, in this case, the major advantage is that the column volume is actually 47 times smaller for the core beads compared to size exclusion. So from a technical perspective, it makes a lot of sense to use core beads. But what about the economical implications for core bead chromatography? In this case, we have looked at the operational costs per batch for core beads compared with size exclusion. We also looked at core beads in standard and single-use chromatography columns. So if we start with a bioreactor size of 200 liter here, you can see that core beads, both in single-use and in stainless steel and in single-use columns, is actually more cost efficient than size exclusion in stainless steel column, as you can see here. And to the right, you can see the same calculation for a bioreactor size of 2000 liter. And as you scale up, you can see actually that the difference is even higher here, and it is in favor of the core bead chromatography, due to that you need quite a lot of buffers and labor when you are using size exclusion in large scale. So what about production capacity? So here you can see the productivity as amount of antigen that can be produced per hour in the same scenarios as before. At both scales, the highest amount of antigen can be produced with core beads in single-use columns. Also in this case, you can see that the difference is more pronounced at a larger scale. And it is of course very important when you develop your process to ensure that you can scale it, but it is also of course important to know that at large scale you have a favorable process economy. So finally, I will now conclude this presentation. And in summary, we have looked at how users of single-use processing report significant economical benefits compared to traditional processing. In the upstream case study, we saw increased profit opportunity with microcarriers in single-use bioreactors due to higher capacity and lower costs. In the downstream case study we have discussed, core bead chromatography can actually enable single-use operation, and it can also be scaled up to large industrial scales. And core bead chromatography also lowers downstream operational costs and increases productivity compared to size exclusion chromatography. So all in all, single-use technology can help to increase profits quite significantly by lowering production costs, but it can also help you to increase production capacity in your plant, and together with this will lead to higher profits. So thank you very much for your attention, and I will be happy to answer some questions. Thank you.
Thank you, Matt. So now we will begin answering questions. If you haven't already, go ahead and type them into the chat window, and we'll get started. So the first question I have is, can the core bead resin be sterilized?
Yes, it can be sterilized. It's quite a robust resin actually, and you can sterilize it, and you can run it in a sort of aseptic mode. So that is possible. That can be quite important actually in many cases when you are purifying a virus that is very large and you have difficulties to sterile filter it, for example.
Okay, so the next question is, how are the microcarriers pre-sterilized? Are they autoclaved or gamma, and how are they delivered in a bag or a bottle?
Yes, so in our case they are gamma treated, and they are delivered in in bottles that can be connected to the bioreactor. So you will have a sort of an easy way to transfer the material into the bioreactor.
Okay. And are pre-sterilized microcarriers commercially available?
Yes, we are about to launch them pretty soon. I'm not sure exactly on when it will be, but we are very close to launch, yes.
Okay, and the next question is, how are pre-sterilized beads loaded into the single-use bioreactor?
So you can do it in different ways, but one way is for example, to weld using tubing and then connect it to your bioreactor, and then you can use, for example, air to move it into the bioreactor. But there are other ways that you can connect it also to your bioreactor. We have tried to develop this product and the sort of bottles in a way that will be very sort of useful for many different types of applications.
And another audience member wants to know, how is the core bead cleaned and sterilized into the column?
Yes, you need to have like a cleaning in place procedure that is sort of common to other types of chromatography resins, and we have cleaning protocols that can be used. If you want to sterilize it, of course you would have to do it. That's a bit more tricky because you have to sterilize the whole column, so you need to ensure that you, for example, use steam in place, which is difficult, of course, with a plastic column. But if you are using stainless, that could be possible. Otherwise, you would have to run it sort of under aseptic conditions.
And so we have another question. Are the results presented for human vaccine studies or for veterinary vaccines?
These are for human vaccines. We have looked into veterinary vaccines also, but we have not presented that today.
Okay. Another question. Do you have examples of scale-up of single-use vaccine process using WAVE as a seed train to inoculate an XDR bioreactor?
Yes, there are actually cases where our customers are using both WAVE and also XDR bioreactors and scaling it up to quite substantial volumes. I cannot actually mention who the customers are, but we do have those cases, and it's becoming more and more popular, of course, to use single-use bioreactors. So we are seeing a sort of increased acceptance all the time, actually.
Okay, and another question. What is the highest BSL safety level for which single-use technology can be used, and can it be applied for BSL-3? And if so, what secondary containment would be required?
Yes, it's a good question. Actually, I mean, the single-use technology as such is not really linked to the BSL level classification, but of course you need to ensure that you are operating it in a way so you have full containment if there is a leakage, for example, of a bioreactor or so. So it is a bit more tricky with very high BSL levels to use single-use, but it's not impossible, and we do see customers and other users that are trying to use this. So that is a good question, but actually, I mean, that is something you have to look on a case-by-case basis, I would say.
Okay. Another question, what are the typical cell densities and virus titers from bioreactor processes?
Yeah, that is a good question also. That is not really related to process economy, I guess, but it's more related to your process. And I mean, you can have everything from quite low yields in terms of numbers of particles. I mean, it can be maybe up to 10 to 6 particles, but we have also seen examples of 10 to 10 or even higher in some cases actually. So that depends a lot on your process, on your cell line. So it's difficult to generalize. But for example, for an influenza vaccine process, maybe you would have a cell-based influenza, maybe you would have 10 to 8 particles per milliliter in a well-optimized process, I would say.
Okay. And we have another sterilization question. Are the pre-sterilized microcarriers animal component free?
Yes, we have 2 versions. So we have Cytodex 1, which is animal component free. So that is mainly used for human vaccines, of course. But we also have Cytodex 3, which is a gelatin-coated microcarrier. And that is obviously not animal component free, but it's used quite a lot in the veterinary industry. And there are also some human vaccines that are produced using this Cytodex 3.
Okay. So we have a number of more questions. We'll try to get through them. So as industry leaders embrace continuous manufacturing, is the current robustness of upstream and downstream single-use technologies capable of meeting the increased usage and run times?
Sorry, I couldn't hear that question.
Okay, that helps. As the industry embraces continuous manufacturing, is the current robustness of upstream and downstream single-use technologies capable of meeting the increased usage and run times?
Yeah, that's a difficult question to answer. I mean, I think it depends on how you implement the technology. Of course, there are some scale limitations in terms of single-use technology. I mean, you cannot run it at any scale. But on the other hand, I mean, most vaccine manufacturing facilities are in the range of the size or the volumes that you can use single-use technologies.
Okay. Are there applications of the core bead chromatography other than virus purification?
Yes, absolutely. I mean, you can use it for other things also. If you want to separate something that is small from something that is large, so you can even use it for cell purification. If you want to purify a cell, you can use core beads. You can use it also for other types of large molecules, and it really depends on the cutoff of the core bead. So, for example, this Captocore 700 that we have on the market is we have a sort of cutoff at 700. So you can also purify very large protein molecules using this, as well as viruses. You can also purify large polysaccharides or even DNA particles.
Can you use it for monoclonal antibodies?
Well, monoclonal antibodies are a bit too small actually to purify using this current Captocore technology that we have, because they will actually enter into the bead.
Okay. And how many times can the beads be reused?
That depends on your feed material and your cleaning procedure. So it's a bit difficult to give like a general description on how many times. But we have pretty good cleaning protocols that we can supply with these core beads, but it depends really a lot on your feed material. Of course, since we have a very strong ligand inside the core beads, you need to use quite harsh cleaning conditions to get to sort of remove the impurities from the beads. But it's possible to reuse it a number of times.
Is there any estimate on what the shelf life of pre-sterilized microcarriers will be?
Yes, we are doing some shelf life studies, so we will have that pretty soon. But we expect the shelf life to be very similar to the standard microcarriers.
Okay, and are there any application notes or white papers on pre-sterilized microcarriers or single-use vaccine production?
Yes, we will launch that also pretty soon. So there will be some application notes and describing how you can use this in a single-use bioreactor.
Okay. And what is the sterility assurance level for gamma sterilized microcarriers?
We will have a pretty well-defined sterilization protocol, and we do have a validation of the sterility and everything, so that will be sort of assured in a very professional way, I would say. And we have, of course, discussed this quite a lot with our large customers using microcarriers, so we are following the sort of industry guidelines that are required.
And we have a follow-up question on the biosafety level. Are you aware of any single-use technologies being used for BSL-3 or higher manufacture?
Yes, it's being investigated quite a lot these days. But as I said, I mean, you need to ensure the containment, and of course, you need to sort of design your plant and also sort of the surrounding equipment very carefully.
Okay. So this question is a little technical. So what is the linear velocity required for proper separation using the core bead, and is the column size so the load represents a percentage of the column volume like SEC columns?
Yes. When it comes to core beads, I mean, you can load, as I showed in the presentation, you can actually load quite a lot of material on your column, and it's also high flow based resin. So I mean, you can actually have quite high sort of flow rates in these columns. So it's a completely different way of running these columns as compared to size exclusion chromatography. But of course, it's very positive that you can reduce the size of the column and also increase the loads.
Okay. Are the cost savings achieved for any vaccines in general, or are they different with different vaccines? And are the savings biggest on influenza protections?
Yes, it's an interesting question. This is something you would have to look for each and every vaccine that you're interested in, I would say. I mean, in these cases, we have looked quite extensively on the influenza vaccine, but also other types of vaccines, actually. And of course, if the manufacturing technology and the cell line and so on is similar, I mean, you would expect similar results, I would say. But there are many different types of vaccine processes, and you would have to look at them, each one individually, I would say.
Do you see the industry looking for single-use bioreactor volumes greater than 2,000 liters?
No, so far, I mean, we have not seen any big interest in larger ones. I mean, theoretically, it's possible to build a larger one, but I think they are quite difficult to handle. And also given the fact that sort of cell culture and the titers and the yields that you get in processes are increasing. So we have not seen any sort of big demand for very, very big bioreactors, single use.
Do you have any data on the reduction of costs for process validation purposes for regulatory bodies?
No, this is not something we have looked into this work, but of course that should be possible to do, I think, also.
And finally, I think this is going to be our last question. What is the typical cost of goods in ratio to the market price for vaccines?
Yes. I mean, as I showed in the first, one of the first slides, actually, you can see that actually more than 50% can be related to manufacturing. And so it's very substantial in terms of cost of goods when it comes to the manufacturing technology. Of course, you have other things also that contribute to cost of goods.
Okay. Well, thank you so much, Matt, and thank you to our audience for your participation and your questions. The Bioprocess Insights webinar series will continue on May 18th with Unlocking the Potential for Efficiency in Downstream Bioprocesses. Today's presentation will be available in the multimedia section of Bioprocess International for on-demand viewing. The previous webinar, How to Improve Productivity and Process Economy Through Intensified Seed Train Strategies, can be found there as well. With that, I'd like to thank you again for joining us, and goodbye.